Chapter 1, Section 8, Synaptic and Neuromuscular Transmission. Types of synaptic arrangements. There are several types of relationships between an input to a synapse, the presynaptic element, and the output, the postsynaptic element. It could be one-to-one, one-to-many, or many-to-one. The one-to-one synapse is illustrated by the neuromuscular junctions shown in Figure 1-16, which is also shown on page 25 of your textbook. A single action potential in the presynaptic cell, the motor neuron, causes a single action potential to fire in the postsynaptic cell, the muscle fiber. And we can see here the release of acetylcholine, which will bind to a nicotinic cholinergic receptor on the muscle fiber, causing it to depolarize and ultimately fire an action potential. One-to-many synapses. The one-to-many synapse is uncommon, but it is found, for example, at the synapses of motor neurons on Renshaw cells of the spinal cord. An action potential in the presynaptic cell, the motor neuron, causes a burst of action potentials in the postsynaptic cells. This arrangement causes amplification of activity. Many-to-one synapses. The many-to-one synapse is a very common arrangement in the nervous system. In these synapses, an action potential in the presynaptic cell is insufficient to produce an action potential in the postsynaptic cell. Instead, many presynaptic cells converge on the postsynaptic cell. These inputs summate, and the sum of the inputs determines whether the postsynaptic cell will fire an action potential. Synaptic input, excitatory and inhibitory postsynaptic potentials. The many-to-one synaptic arrangement is a common configuration in which many presynaptic cells converge on a single postsynaptic cell, with the inputs being either excitatory or inhibitory. The postsynaptic cell integrates all the converging information, and if the sum of the inputs is sufficient to bring the postsynaptic cell to threshold, it will then fire an action potential. Excitatory postsynaptic potentials. Excitatory postsynaptic potentials, or EPSPs, are synaptic inputs that depolarize the postsynaptic cell, bringing the membrane potential closer to threshold and closer to firing an action potential. EPSPs are produced by opening sodium and potassium channels, similar to the nicotinic cholinergic receptor. The membrane potential is driven to a value approximately halfway between the equilibrium potentials for sodium and potassium, or about 0 millivolts, which is a depolarized state. Excitatory neurotransmitters include acetylcholine, norepinephrine, epinephrine, dopamine, glutamate, and serotonin. Inhibitory postsynaptic potentials. Inhibitory postsynaptic potentials, or IPSPs, are synaptic inputs that hyperpolarize the postsynaptic cell, taking the membrane potential away from threshold and farther from firing an action potential. IPSPs are produced by opening chloride channels. The membrane potential is driven toward the chloride equilibrium potential, which is approximately negative 90 millivolts, which is a hyperpolarized state. Inhibitory neurotransmitters are gamma-aminobutyric acid, or GABA, and glycine. Integration of synaptic information. The presynaptic information that arrives at the synapse may be integrated in one of two ways, spatially or temporally. Spatial summation. Spatial summation occurs when two or more presynaptic inputs arrive at a postsynaptic cell simultaneously. If both inputs are excitatory, they will combine to produce greater depolarization than either input would produce separately. If one input is excitatory and the other is inhibitory, they will cancel each other out. Spatial summation may occur even if the inputs are far apart on the nerve cell body, because EPSPs and IPSPs are conducted so rapidly over the cell membrane. Temporal summation. Temporal summation occurs when two presynaptic inputs arrive at the postsynaptic cell in rapid succession. Because the inputs overlap in time, they summate. Other phenomena that alter synaptic activity. Facilitation, augmentation, and post-tetanic potentiation are phenomena that occur at synapses. In each instance, repeated stimulation causes the response of the postsynaptic cell to be greater than expected. The common underlying mechanism is believed to be an increased release of neurotransmitter into the synapse, possibly caused by an accumulation of calcium in the presynaptic terminal. Long-term potentiation occurs in storage of memories and involves both increased release of neurotransmitter from presynaptic terminals and increased sensitivity of the postsynaptic membrane to the neurotransmitter. Synaptic fatigue may occur where repeated stimulation produces a smaller than expected response in the postsynaptic cell, possibly resulting from the depletion of neurotransmitter stores from the presynaptic terminal. Neurotransmitters. The transmission of information at chemical synapses involves the release of neurotransmitter from a presynaptic cell, diffusion across the synaptic cleft, and binding of the neurotransmitter to specific receptors on the postsynaptic membrane, all to produce a change in membrane potential. The following criteria are used to formally designate a substance as a neurotransmitter. The substance must be synthesized in the presynaptic cell, the substance must be released by the presynaptic cell upon stimulation, and if the substance is applied exogenously to the postsynaptic membrane at physiologic concentrations, the response of the postsynaptic cell must mimic the in vivo response. Table 1-4 illustrates a number of different neurotransmitters and how they can be grouped into different categories. There's acetylcholine. There are multiple biogenic amines such as dopamine, epinephrine, histamine, norepinephrine, and serotonin. Several amino acids such as gamma-aminobutyric acid, otherwise known as GABA, glutamate, and glycine, and multiple neuropeptides listed on the right on Table 1-4. Acetylcholine. The role of acetylcholine as a neurotransmitter is vitally important for several reasons. Acetylcholine is the only neurotransmitter that is utilized at the neuromuscular junction. It is the neurotransmitter released from all preganglionic and most postganglionic neurons in the parasympathetic nervous system and from all preganglionic neurons in the sympathetic nervous system. It is also the neurotransmitter that is released from presynaptic neurons of the adrenal medulla. Figure 1-17, which is shown on page 26 of your book, illustrates the synthetic and degradative pathways for acetylcholine. In the presynaptic terminal, choline and acetylcholine combine to form acetylcholine, which catalyzed by choline acetyltransferase. When acetylcholine is released from the presynaptic nerve terminal, it diffuses to the postsynaptic membrane where it binds to and activates nicotinic or muscarinic cholinergic receptors. Acetylcholinesterase present on the postsynaptic membrane where it degrades acetylcholine to choline and acetate. This degradation terminates the action of acetylcholine at the postsynaptic membrane. Approximately half of the acetylcholine that is released from the degradation of acetylcholine is taken back into the presynaptic terminal to be reutilized for synthesis of new acetylcholine. Norepinephrine, epinephrine, and dopamine. Norepinephrine, epinephrine, and dopamine are members of the same family of biogenic amines. They share a common precursor, tyrosine, and a common biosynthetic pathway shown in Figure 1-18. These are known as the catecholamines. Figure 1-18 is also shown on page 30 of your book. Tyrosine is converted to L-DOPA by tyrosine hydroxylase, and L-DOPA is then converted to dopamine by DOPA decarboxylase. If dopamine beta hydroxylase is present in the nerve terminal, dopamine is converted to norepinephrine. If phenylethylamine N-methyltransferase, also known as PNMT, is present, with S-adenosylmethionine as the methyl donor, then norepinephrine is methylated to form epinephrine. The specific neurotransmitter secreted depends on which portion or portions of the enzymatic pathway is present in a particular type of nerve or gland. Thus, dopaminergic neurons secrete dopamine because the presynaptic nerve terminal contains tyrosine hydroxylase and dopa decarboxylase, but not the other enzymes. Adrenergic neurons secrete norepinephrine because they contain dopamine beta hydroxylase, in addition to tyrosine hydroxylase and dopamine